Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Dr. David Miller, Vice President of Cheminformatics, will discuss the new features of our flagship machine learning platform, ADMET Predictor. Eric Jamois, Senior Director of Business Development, will be our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. And I'll pass things over to Eric.
All right. Thank you, Jasmine. So good morning, good afternoon, and good evening, everyone. Thank you for joining us in this webinar featuring our most recent release of ADMET Predictor. We are going to focus on the new 11 release. There is a significant amount of new functionality and enhancements in this release, which David will be covering in detail. The highlight is, of course, the new pKa ionization model, which you may have heard about, was developed in collaboration with 4 commercial companies. It's a huge amount of work that's gone into this, so we're very proud to bring it to market. I'm also reminding all of our attendees that our MedChem Studio module remains free with any paid-for module. So if you haven't gotten your license, your free license yet, make sure you reach out to us and we'll get it over to you. But I don't want to steal the thunder from David. So without any further ado, it's my pleasure to introduce Dr. David Miller, Vice President of ADMET Cheminformatics R&D at Simulations Plus. David, the floor is yours.
Okay, thank you, Eric. And again, welcome everyone to today's release webinar for ADMET Predictor version 11. Just a couple more quick housekeeping items. Let's see, advance the slides here. I was told that I should at least include a picture of myself since I don't have the camera on. So this is from up here in the sunny Bay Area in Northern California. And then second, unlike in previous release webinars, today I'm going to have to skip the basic introduction to Predictor and instead jump right into the new functionality because we have quite a bit of ground to cover. I would just encourage any newcomers to visit our website and watch some of our past webinars, as well as some of the helpful training videos. Okay, so as Eric just mentioned, the flagship feature for this new release is the improved pKa model, which has been rebuilt using a much larger quantity of experimental ionization data. The training set now incorporates proprietary data received from 3 new industrial partners, identified for now at least as partners 1, 2, and 3. They have provided respectively 19,000, 2,400, and 4,100 compounds. So the total number of ionization constants has now more than doubled from around 33,600 to over 70,000. So this latest work is continuing our successful track record of industrial partnerships that originated back in 2011 when Bayer Pharmaceuticals contributed around 17,000 proprietary compounds. That was about 20,000 ionization constants. And I'll just remind everyone that the pKa model resulting from that earlier collaboration was already ranked as the top performing QSAR-based model in the Sample 6 pKa challenge. You can find that publication in the Journal of Computer-Aided Molecular Design from 2021. So we believe that this improved model will keep us well ahead of the competition. These plots show the chemical space of the various data sources of the new model in the form of principal components graphs using predictors, molecular and atomic descriptors. The blue and the dark green represent the public data plus the bare data that we had prior to AP11. And then the remaining three colors represent the new data. So you can see that while there is some overlap, we've definitely expanded the chemical space overall by adding this new data. The expansion is even more compelling in video form, and I believe we have that up on our website, or will soon. The overall performance plots for the prior AP 10.4 and updated model in AP11 are shown here. Even though the size of the data set has more than doubled, the performance has actually improved, both for the training set and for the external test set. These two figures are taken directly from the respective user manuals for AP. The improvement is more dramatic if you look only at the new data. Here focusing on the largest set from partner 1. This is roughly 30,000 ionization constants. Predicting these data using the prior 10.4 model yields an RMSE of about 0.79, but that drops down to just about 0.41 with the new model. So a very satisfying overall improvement. Some final bullet points on the new pKa model. Our modeling descriptors related to ionization have been updated. So most ADMET Predictor models have been retrained. So you can expect some small differences in the predicted values. Existing user models, so those built with our ADMET modeler software, should similarly be retrained if they incorporate any ionization descriptors. Simulations Plus can assist you with this effort. We have some command line utilities that can help streamline the process. And then finally, look for a future webinar focused on the pKa model development with partner companies participating. There are some complexities involved in arranging this, but we're hopeful that we can put it together in the very near future. ADMET Predictor 11 also includes new and improved models for CYP inhibition. First, there are new regression models for the quantitative prediction of inhibition constants, or Ki values. We have seven separate models for the isoforms listed here, 5 major and 2 minor. The data sources include the GoStar, DIDB, and Kemble databases, as well as individual publications. There are also new and updated classification models that predict yes, no, or inhibitor, non-inhibitor. The new models are for these 4 minor CYP isoforms, and then we've updated the existing models for the 5 major ones. Here, the data sources include the PubChem database, as well as the data that we curated in the development of these Ki models. The thresholds delimiting inhibitor, non-inhibitor were less than 10 micromolar and greater than 57 micromolar for the PubChem data. That data was already classified on the basis of measured AC50 values. And then for the curated Ki data, less than 10 micromolar and greater than 30 micromolar. And then just as a technical note, the classification models serve as a filter for the quantitative Ki models. So if the former predict no, the latter will predict non-inhibitor rather than a quantitative Ki value. That follows the same convention as several of our other metabolism and transporter models. This chart just updates the status of our current SIP models, which include substrate, inhibitor, and sites of metabolism among the classification type models. And then for the quantitative regression models, the new KI models, as well as KM, Vmax, and intrinsic clearance. The yellow indicates the missing models, which are for minor SIPs, where we were not able to obtain enough high quality data. For the models that we do have, the performance statistics and descriptions of the data and curation protocols are all contained in the user manual. All right, switching from predictive models to cheminformatics features, we've added the capability to perform 3D similarity screening. So this functionality is made possible after the introduction of 3D conformer generation in ADMET Predictor 10.4. So this is a new virtual screening tool based on a combination of 3D shape and pharmacophore feature similarity. The idea is to find compounds similar to one or more 3D reference conformers. The screen is typically against a large 3D conformer database that's generated from compounds available internally or commercially. And database hits with a high similarity to the reference would have a higher chance of being active and would be prioritized for acquisition and testing. This slide just illustrates the idea. This is from a 2005 publication on one of the earliest 3D shape tools, rocks from the company OpenEye. The idea was to find hits similar to this known ligand for which there was an existing protein ligand X-ray structure. So ROX was used to screen a large 3D conformer database, and many of the compounds with high 3D similarity scores turned out to be active. The active shown here had its crystal structure determined, and this image shows the two ligands superimposed in the active site. What's particularly noteworthy is that ROX was not only able to predict activity pretty well, but in several cases like this one, it was able to quite accurately predict the binding pose as well. So this was an early demonstration of 3D shape matching. And since then, the functionality has become very popular, with many believing that shape screening is competitive with, or even superior to, full-blown 3D docking. So briefly on the methodology as implemented in ADMET Predictor, the similarity scores consist of a shape term that's based on overlap volume, and then optionally a feature term based on the alignment of pharmacophore features. And users can adjust the relative weights of those two terms. This is a screenshot from Predictor where you can define and visualize the pharmacophore features. By default, there are H-bond donors and acceptors, positive and negative charges, and rings. And then within Predictor, there's a query syntax that allows you to define your own features. The overlap volume is computed using atom-centered Gaussians. So this approximates the exact hard sphere overlap, but is faster and more convenient for gradient-based optimization. So there was a paper from 1995 where this functional form was introduced. The exact hard sphere overlap was approximated using a sum of Gaussians, and then further simplified in such a way that you only need to consider all pairs of atoms. It was shown that this simplification still yields a volume that's quite close to what you would get exactly, but orders of magnitude faster, and as I said, suitable for optimization methods. So then the overall protocol would be, given a query conformer and a single database conformer, you would, for each of several starting alignments, use an optimization technique like BFGS to find the rigid alignment that maximizes the similarity score. Again, volume plus features. So we evaluated the performance of the tool using the freely available DUD data set, which is commonly used to evaluate 3D virtual screening methods, not just similarity based, but also docking, for example. So the DUD set consists of 40 target data sets that have both actives and inactives. And there are 3D structures included, either X-ray or from software. So the testing protocol would be that for each of the 40 targets, you would create a 3D conformer database using ADMET Predictor. So that's new functionality in AP11. Then perform a 3D similarity screen of each active against the target database, and then compute average ROC curves over all actives in the corresponding average AUC, the area under the ROC curve. And I'll explain these curves in a second. The best predictor results, an average AUC of close to 0.8, is quite competitive with other 3D virtual screening tools described in the literature. So this is showing 5 of the 40 average ROC curves. It's ordered from best to worst, evenly sampled among the 40. So for those unfamiliar with ROC curves, you're measuring the fraction of actives that you see as you look at the compounds in order from highest to lowest 3D similarity. So here you would have an almost perfect result where the highest similarity compounds are all active, and the resulting AUC is very close to 1. And then in contrast, looking at this worst example, you're not doing that much better than random, with an AUC much closer to 0.5. Similar results are found with the other published similarity methods, where some targets perform really well, and then others are much, much harder. But overall, an average AUC of nearly 0.8 across all 40 targets is very competitive, at least as far as we're able to tell from the literature, not being able to actually run the other packages ourselves. This evaluation used the default set of pharmacophore features and a weight of 10 for the feature term relative to the volume term. We've also included a GPU implementation of 3D similarity screening, which greatly accelerates the performance. You can see the options window here includes a GPU checkbox, and that will be enabled if a compatible graphics card is detected. And for now, that has to be an NVIDIA card. And then this just describes a simple timing test that was run on a laptop with the specification shown here. So using a single COX-2 inhibitor as the reference against a 100 million conformer database of chemical compounds. On the CPU, the screen takes about an hour and a half, using all 16 available CPU cores. And then on the GPU, it's well under 2 minutes. And that 50 to 60-fold speed up is what we've observed across multiple examples, though obviously that would depend on the hardware that's being used. And then finally, this just shows the scores generated for this example using the CPU versus the GPU. They're very close. The RMSE is just 0.001. You do find that the small numerical differences can lead to slightly different solutions in some cases. So that new 3D similarity feature, in addition to being a standalone tool, is now also integrated into our AIDD, or AI-driven drug design module. So AIDD, just briefly, is an iterative protocol based on molecular transforms and Pareto optimization that's used to discover new compounds simultaneously optimized across multiple target objectives that are defined by the user. So now in AP11, when selecting the target objectives, in addition to ADMET properties, the high throughput pharmacokinetic endpoints like fraction absorbed, activity models, and so on, you can also include similarity to one or more 3D reference conformers, those conformers presumably being active against the target of interest. So the way it works is that in each AIDD generation, the new virtual compounds are converted to a 3D conformer database and then screened using the one or more 3D reference conformers. I'm going to try to show a quick demonstration of this after the slides and hopefully explain it a little bit more slowly and clearly. In just the last 3 months or so, we've entered into 2 new collaborations around our AIDD technology, where we are aiming to design novel compounds with good properties around ADMET, PK, and activity. In both of these projects, existing lead compounds are available with known 3D structure. So we've been able to make use of this new 3D similarity functionality. And so far, it's looking to be a very useful addition. We hope to get some initial experimental results from these collaborations in the next few months. So stay tuned for news on that front, and hopefully 1 or 2 dedicated webinars as well. We've made several enhancements to the new REST API based on user feedback. So the run command request, which is used to simulate predictor command line runs on the server, now supports what we call script file workflows, which extend the functionality beyond just ADMET property prediction and HTPK simulations. The new workflows include metabolite prediction, log-D and solubility versus pH profiles, 3D conformer generation, and several others. The predict ADMET request now supports a new parameter that triggers the use of our 3D predictive models where available. The get status request can now be submitted without a job ID, in which case the response will indicate which jobs are currently running, complete, or in the queue. And there's a new configuration parameter that allows users to control whether the licenses are checked out immediately when the service starts up, which is the default, or only as needed for each individual request. All of these and other enhancements are explained in the separate REST API user manual. And this just shows some example result images generated for 2 of the new API workflows. This one, a prediction of CYP3A4 sites of metabolism, and then this one, the prediction of metabolite structures. This is very similar to what you see in our MedChem Designer software. And then I'll just briefly highlight a few other enhancements in this version. There are some new command line workflows related to the 3D similarity functionality. One for creating the 3D conformer databases, and a separate one for exporting them to an SD file, and then a workflow for performing the 3D similarity screens against those databases. There are some improvements to 3D coordinate generation, specifically the handling of macrocycles containing multiple cis-trans stereocenters. There are new workflows, both in the graphical interface and the command line, for adding and removing 3D hydrogen coordinates. And there is a new advanced 3D conformer option, allowing you to augment the torsion angle preferences using custom smarts patterns. AIDD supports the use of our 3D predictive models. This does require that all of the so-called seed compounds have 3D coordinates defined. Opening large SD and SMILES files has been made much faster by exploiting multi-core CPUs. The assignment of PK50 values, these are individual dissociation midpoints has been improved. This is some interesting work done in collaboration with one of our large customers. And my colleague, Robert Franskevich, who was also responsible for building the improved pKa model, he's finishing up a publication that describes this work in detail. And then there are several other enhancements that you will find described in the README file and the user manual. So I think at this point, I'm going to stop and we'll, I think, have some time still to run some quick demos with the new version. But I believe we are going to pause for an instant and take another poll.
Thank you, David.
Thank you, David. I'm going to go ahead and launch the second poll. And I'll give everybody a few seconds to answer these two questions as well. See some answers coming in. Couple more seconds. All right, I'll go ahead and end it there. Thank you.
Okay, thank you. So I'm going to switch over and actually launch the soon to be released ADMET Predictor 11. On the release date, we're just wrapping up some finishing touches to the installer itself. Just making sure that the install goes smoothly on Windows 11 in particular. But we anticipate that being done in the next several days. And then that will shortly be followed up by the release of the Linux version of Predictor. So in the next week, I think you can expect both of them to be out and ready to be downloaded. Okay, so first, I'll just very briefly show how fast and easy it is to generate our predicted ADMET properties. So I'm going to open up a set here. This is a set of 1,000 compounds from the Chembl database. So if I click the Add Me button, people are probably pretty familiar with this already. You see a list of the available categories of predicted properties. So it includes physicochemical, transporters, metabolism, toxicity, and modeling descriptors. If I just click the calculate button, you see even for 1,000 compounds, it's extremely fast. It does, of course, make use of the multiple CPU cores. All of the predicted properties are displayed. And then what I really wanted to show here is that in addition to the improved pKa model, that in the pKa's are shown here in these three highlighted columns, that those improvements do carry over to the more advanced pKa microstates display, as well as to the corresponding ionization graphs shown here. My colleague, most likely Robert, will discuss these things in much more depth during the future pKa model webinar that I mentioned earlier. So I am going to leave this discussion for that later time. Okay, so the next thing I want to show is just talk briefly again about the new 3D similarity searching feature. So here I've opened up an SD file that contains 3D coordinates. So in Predictor, you'll always see the 2D structure displayed, but in the interface, there is an indication that the 3D coordinates have been loaded and stored. And then if you have linked a 3D viewer application with Predictor, you can select the compound and then launch that viewer and see the 3D coordinates. shown here. This one happens to have the hydrogens removed, although that's of course not necessary. So this happens to be a known COX-2 inhibitor. This is from the DUD data set, and I believe it originates from a patent. I think the company is Merck, if I'm not mistaken. So in this example, imagine that we want to use this 3D structure to screen a 3D conformer database. So first in Predictor, obviously you can create those databases either from the GUI or, as I mentioned, from the command line. It's a straightforward process. You just select an SD file or a SMILES file, and then some parameters related to the conformer generation, like how many conformers per compound and energy window, RMSD between conformers, and so on. That's all described in the user manual, and we talked about that in the prior AP10-4 webinar. I'll just briefly show what the database would look like. So once you generate it, you end up with a folder containing a series of files. So they're binary files. Each one contains roughly 1 million 3D conformers. So there's 10 files here. So that's about 10 million conformers. It occupies about 3 gigabytes on the disk. So it's not trivial. These files are totally independent of one another. So you could distribute the creation of a 3D conformer database among several different machines, and then combine those files later however you want. And then, similarly, if you're doing a 3D similarity screen, you can distribute that among several machines as well, right? Because these files are totally independent. Okay, so this is just a sample database with 10 million. So it's 1/10 of the size of the one that I had mentioned earlier in the slides, because I have to be able to screen it quickly since this is a short demo. So to launch that screen then within the interface, there's an option under 3D search by similarity. I've already filled out options here. So I've selected that folder that I just showed. So it indicates the number of conformers that are contained in that database. So it's just over 10 million. Then an output file for results. I have selected the option to run this on the GPU. So this particular laptop does have a GPU that's from NVIDIA. I'm using a feature weight of 1. So it's equal weighting of the pharmacophore feature component of similarity relative to the volume component. And then I've selected just the default set of pharmacophore features that we provide. And if you click a button here, you can see the reference compound that I'm using shown here with the pharmacophore features highlighted. So here there's only hydrogen bond acceptors and then the ring centroids. And then again, you can use the built-in query syntax within Predictor to define your own features, that perhaps capture some important motif that's particularly relevant to binding to your particular target, for example. Okay, so if I run this, you'll see that it's quite fast. So for each of those 10 million database conformers, it's doing multiple BFGS optimizations because it's using lots of different starting points. So at least with the GPU option, it's extremely fast. When you get the result window, it contains all the database hits with a similarity greater than a threshold that was specified when you initiated the run. And I chose 0.8. I failed to mention that the similarity is returned in the form of a Tanimoto score, which is going to vary from 0 to 1. So all these hits have a Tanimoto similarity of at least 0.8. Some of the hits you can see have a motif that's very similar to the reference compound with the sort of central atom here. So that's not surprising. But then there are some that are, quite different. And this one, everyone probably recognizes as the more classical COX-2 inhibitor motif with the central ring and the ortho-substituted aryl substituents here. I believe this actually is the the Vioxx compound, Chembl 122. In the output file, the best alignment, so the best alignment between the hit and the reference, the one that yielded the displayed similarity, is saved as a separate SD file, and it's included as a link here. And I've associated SD files with a separate viewer program. So if I click on that, it'll launch, in this case, Biovia viewer, so you can see the overlay. So this is the starting reference compound, and then this is what the overlay looks like. So again, the Vioxx compound. So the overlay is, pretty compelling to my eye. The overlap is good, and the alignment of the pharmacophore features as well. And I think what's interesting and what's somewhat different than the rocks example from the slides is that here, I don't think you would expect a purely 2D similarity method to pick this compound up as a particularly high scoring one, just given the differences in the ring connectivities here. So it kind of illustrates the virtues of 3D similarity screening as opposed to 2D. Okay, so now I just briefly want to show what that would look like inside of an AIDD run. AIDD is quite a bit more complicated. So I mentioned it would probably be valuable to go back and look at some of the prior webinars that we've given on AIDD. My colleague, Jeremy Jones, I gave one, I think just last month, that was related to one of those two collaborations that I mentioned. But I'm just going to kind of run through it really quickly, just to give an idea of the integration of the 3D similarity. So imagine again that we're interested in designing COX-2 inhibitors. So in this case, we're going to be using as one of the objectives 3D similarity to that same reference compound that I used before. So that's shown here. So in AIDD, you start with a seed compound, and it's typically a fragment. So I've chosen a small fragment here. It's actually from this compound. So I select that, launch AIDD. I've already selected my objectives, and there are going to be 4 of them. 3 of them are shown here. Synthetic difficulty is a scoring function that's meant to keep the new compounds from becoming too complex. The idea being that we would like to be able to synthesize them. And then we're going to run the high throughput pharmacokinetic simulations to estimate fraction absorbed. And then finally, ADMET risk, which is our composite score that includes a lot of individual ADMET properties into a single number. So we're minimizing synthetic difficulty, maximizing fraction absorbed, and minimizing ADMET risk. And then the 3D similarity is activated using a separate option. So we've implemented this by requiring that you edit a text file that contains various parameters related to the run. So for example, the path to the SD file containing the 1 or more 3D reference structures. And then there are parameters related to the creation of the 3D conformer databases and the 3D similarity screen. So that's the mechanism that we've chosen, at least for now, for integrating the 3D similarity scores. Okay, then just briefly in AIDD, you can supply a scaffold. So you are restricting all the new compounds to have a certain overall motif. And here we're basically just ensuring that the starting fragment is preserved in all of the compounds. And then furthermore, that no substitutions are allowed at these particular positions. So we just want to grow this fragment in this direction out here, kind of like this compound. And then finally, there's a lot of parameters here that you can learn about elsewhere, but I have to run it for just a very short time, 5 generations, just to be able to finish it in a reasonable amount of time. I'll start running it and just mention that in this case, the slow step is the generation of the 3D conformer databases. And you can kind of see that in the progress window here. So, but it still runs quite quickly. It's on the final generation. And then I just want to show what the layout looks like in the output file. So you get a set of new compounds here. Some of them are not very evolved because I only ran it for just a very small number of generations. But you see that it was able to find some compounds with the more classical COX-2 inhibitor scaffold that we saw before. If I sort just to look at the most 3D similar compound and show that link. So this is another pretty well-known COX-2 motif with now a fused system here in the central portion. I'll just really quickly click on that. Okay, so again, reference and then the alignment. So a little bit different than before, but still a pretty compelling overlap. Okay, I think that's about all I wanted to say about AIDD. Just a reminder that we do anticipate some results from those collaborations, and we'll hopefully schedule some new webinars to talk about those results.
All right.
Okay, I don't know, Eric, I think we've got a couple minutes. Can I show one last thing?
Sure, yeah.
One last thing, okay. This is sort of interesting. So this is on the API. So the API is something we recently introduced to make it easier to access our ADMET Predictor functionality from third-party software. In the past, I've demonstrated the API using what you would call a thick client. So a full molecular spreadsheet kind of meant to mimic something like Live Design or D360. But today I wanted to highlight a piece of free software. It's called Visual Studio Code. And it lets you create Jupyter notebooks and run Python scripts. And even for a non-Python person like me, it's pretty straightforward to write code to make API requests. So just going to walk through it really, really quickly. I believe, looks like have the ADMET Predictor service running locally on the laptop here. So I can make requests to localhost in a particular port. So this is just the predict ADMET request, sending in a single smile string. This is JSON format, pretty straightforward. Request for 3 different properties: log D, solubility, blood-brain barrier, classification model. So I can just run it directly within this. It should ping the server, get a job ID back, and the properties. If I were a better programmer, I could probably display these in some kind of graph or something. But you see, for log D, I think you just get a value. But for solubility, it's the value plus an uncertainty prediction. And then for the classification model, the blood-brain barrier, it's a prediction and a confidence estimate. We've talked about that many times before. And then now one of the new workflows that I talked about, the so-called script file workflow using this run command. So here we're passing a ADMET Predictor command line argument, and here it's the name of a file. And then the contents of that file, so-called script file, are included in the JSON. So this one is for the creation of atomic property images. One of the parameters in that file is a smiles file. So the contents of that smiles file are also included in the JSON. Same smiles as I used before. And then as before, there's some more code here where you have to actually wait for the job to complete because it, possibly enters a queue because there could be other jobs running. And then when it's done, you can request the results from the server, in this case, an image, and then display it right here within the Jupyter notebook. So if I run that, again, it's fast, indication of success and the name of the file that was generated on the server. And then you just grab that file and pull it back down, and it can be displayed right within this, this code window here. The images that are actually created don't have these weird axis labels here. That must be a Python thing, I guess. And then finally, the last one, very similar, metabolite prediction. So it's exactly the same, except we're using a different script file. So slightly different parameters. But again, we submit the job, wait for the completion of it, and then grab the result file and then display it. So again, this is very similar to what you would see in our MedChem designer. So hopefully that explains some of the new API functionality and provided some ideas on how you might actually be able to interact with the API. All right, so I think Eric is chomping at the bit here. So I'm going to turn it back over to him and let's do some Q&A.
Yeah, good stuff, David. Thank you. Thank you. So now we've got a couple questions that popped up in the Q&A pane that we can go through. One of them relates to the access to the new release. I think you mentioned, about a week or so for the Windows version to be available through ShareFile. What about the cloud access for the academic users who are on the University Plus program? Within what timeline do we expect that we'll have the new version available through this mechanism?
I would have thought that might be more in your wheelhouse, Eric, than mine. I guess I'm not sure. I'm assuming it would not take long at all.
Yeah. I mean, I think when the release is available, I mean, it's really not very complicated for us to, put it up on the cloud. So, I would expect that to be up within days, literally. So yeah, it won't take long. And the other one, that one I don't know, is there a GPU acceleration available in the cloud access? I
don't know how the cloud access works. So the question implies that the software actually runs in the cloud. Is that right?
Yeah, it does. I mean, it does. It's a login that is, provided to the University Plus users.
Okay. I'm not sure about that. Yeah, it very well may not. This came up recently, actually, in our company that, it's a virtual, instance typically, right, in the Amazon cloud. And I think I heard a rumor that with virtualization, there's sometimes an issue with sharing all of the hardware resources. And so I'm not sure that you would, even if the, physical machine had a GPU, I'm not sure that the virtual instance would have access to that. I'm not an expert on hardware by any means. So, we'll have to look into that and see.
Yeah. I mean, I think these machines are, typically compute servers. I mean, they're Windows servers. I don't think they have, graphics capabilities of on their own.
Yeah, it could be something that, we could specifically request, though. So we'll look into that.
Yeah. I mean, it's an interesting question. That's my guess, but to be confirmed. So another question for the 3D screening. Why is it necessary to generate a conformal database rather than using a 3D alignment algorithm? So I imagine, our users are referring to certain forms of flexible alignment where you're essentially generating conformers on the fly. I think it's just a question of approach, right? I mean, it's just
it's a good question, and it actually, gets to something that we are currently discussing, which is a alternative approach in which we're doing things more dynamically. There are a lot of other tools out there, that use the conformer database approach. And so that was the first one that we chose, but we are, looking into other options as well, that would be more dynamic alignment, that can handle, like flexibility and stuff. So stay tuned.
Yeah. Here's another interesting question on the API. That one just came up. Is the API functionality available through the cloud access will it be possible to make a request via VS Code or Jupyter Notebook on the cloud version? I don't know. I mean, to me, it seems it's not really so much of a technical limitation as is how comfortable, users in general, and especially our commercial users, would be in, sending, confidential structures to a cloud machine, unless it's their own, private, cloud server that they are contracting through, AWS or some other provider themselves. That to me would be the main culprit. I
And
don't know. Do you have a different read on it?
I guess as far as the University Plus program goes and the API, I'm not sure. And unfortunately, I'm not that familiar with that program and how we, provide predictor in that scenario. But I just wanted to clarify one thing with regard to the API, that in a typical case at a company, the ADMET Predictor server is at your site within your firewall. I don't think I mentioned that. So just to clarify that, in the typical case at a company who's licensed ADMET Predictor, there's no sending your structures out over the internet. It's all within your own firewall.
Yeah. No, that's a really important point, and we get that question really often. And I think our commercial users especially are very sensitive to the fact that ADMET Predictor runs behind their firewall, and there is no data that is leaving their facility, being processed elsewhere and brought back. Everything happens within their walls. And again, if they choose to put our software on, on their cloud, it is, the dedicated cloud machine that they have contracted and that they are responsible for in terms of security. So, it's fully managed by our clients in that respect. So that's also an interesting question here that and that comes up fairly often. For the prediction of new molecules when using ADMET Predictor QSAR models, would it be possible to get the applicability domain information, preferably Tanimoto's similarity for the molecule? So maybe you can comment on that. I mean, we do provide the out of scope, out of scope flags for predictions, and that is based on, I believe, what was once called OPS, optimum prediction space, but basically relating to the descriptors that are involved in the model. So if maybe you want to say a little bit more about that, or I don't know if I fully answered the question, but
yeah, it's something that we have talked about quite a few times in the past. People have asked for a range of different things, everything from maybe just a Tanimoto score, which I think seems reasonable, to, an actual picture of, what is the most similar compound in your training set to the one that I'm asking for predictions for. That second one is more problematic because, we don't even have the rights to show some of the structures in our training sets. We've talked about, the similarity one a few times. I think it's something that we should consider again as a feature. So just to clarify, I mean, right now, as Eric said, you get a flag indicating whether the compound you're predicting is outside of the so-called applicability domain, which is based on our molecular and atomic descriptors, but it is basically a yes, no. I mean, yeah, I mean, with the caveat that you also, for many of our models, get a quantitative uncertainty estimate as well as the confidence estimate for the classification models.
Yeah, and you can also do the descriptive sensitivity analysis to see how far outside that particular descriptor that was flagged is. So are you a little bit outside, and how sensitive, how important is it in the model relative to the other descriptors that are involved in that model? So you can know two things: how sensitive the model is to this descriptor or these descriptors, if there are multiple ones that were out of scope, flagged out of scope, and how far out of scope they are. So you can get sort of two clues where, on the reliability of that prediction. So for now, that's what we output, and it's pretty explicit in the software when it's underlined red that the prediction is so-called out of scope. All right, hopefully we answered that one. So this is something that we often get to. Are your models validated for the purpose of replacing regulatory animal tests for determining toxicokinetics and pharmacokinetics? So we can start answering it. It's actually a pretty loaded question. You can take the first stab, or I can take the first stab, David.
Yeah, there was some legal language in that question that I'm not sure I'm qualified to really address. We deal obviously on the services side of our business with regulatory agency interactions quite a bit. I would probably want somebody from that group to maybe chime in here on that. I mean, obviously GastroPlus does use predictions from ADMET Predictor, so it's not as though companies who are making these submissions, it's not like they don't use ADMET Predictor. But as to whether predictions themselves can serve as replacements for things that ordinarily would need to be measured, that could be a little outside of my domain.
Yeah, I know that for the mutagenicity models, we have generated the QMRF forms that are requested, typically, that do disclose the construction, many of the details that are required. And again, we can provide these QMRF forms upon request. On the toxicokinetics side, I actually came across an interesting paper from the FDA Office of Food and Nutrition, if I remember correctly, that makes use of our HTPK technology to look at toxicokinetics. So in other words, turning HTPK into HTTK, high throughput toxicokinetics, for a certain class of compounds. So there have been some uses that have been made in that space. So I'm not providing really a direct answer, but pointing in a couple directions. And I think they were anthraquinones, if I remember correctly. So I don't think we have a whole lot of time left. I think we've taken most of the questions here. Maybe one last one. I think we have 1 minute left. Would the pH-dependent log-D profile as well as PK microstates be available through the API in the new version?
The log-D, okay, say that one more time. The log.
Yeah, would the pH-dependent log-D profile as well as PK microstate be available through the API?
The first, yes, but the second, as far as I just briefly showed what we call the pKa microstates display, where you kind of see on the y-axis, the pH, and then you see, the various ionization microstates laid out nicely. That is something that currently you can generate neither from the command line nor from the API. But that is something that we very much hope to address in the next version. So that one, that pKa microstates display is currently Windows graphical interface only. The log-D or solubility versus pH profiles, that you can generate from command line, from API. So we're getting there.
Yeah, well, we keep adding more at each release. We're trying to put a little bit more functionality on the API and the rest of the software as well. And I think everybody can appreciate that. So, well, Thank you, David. Thank you to everybody who joined us today. Well, we hope everybody found this interesting, and certainly we look forward to having the releases, the new releases, in as many new hands as possible.
Thank you both. Thank you, David. Thank you, Eric. For any questions that we were unable to answer live today, our BD team will reach out to you with answers. You can also visit our website to learn more about ADMET Predictor and our other products and services. We also have another webinar next month about AI, ML, PBPK, and QSP, QST modeling in drug discovery and development. We're going to talk about how modeling can support better decision-making and streamline the entire drug discovery and development process. Again, you can register on our website, simulationsplus.com/events. And I also left some links in the chat for more information on the University Plus program. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you again.
Thanks to everyone. Bye.
Thank you.